Welcome. Today, we're diving into the topic of analytical method transfer between laboratories. We'll explore how to ensure consistency and reliability when moving testing methods from one lab to another. This process is crucial for maintaining quality and accuracy across different locations. Let's get started. Let's start by understanding what analytical method transfer, or AMT, really means. It's a documented process that shows an analytical procedure works just as well when moved from one lab to another. The goal here is clear: to ensure reliable results, support site transfers, confirm lab capability, maintain product quality, and meet regulatory requirements. Typically, this involves a sending lab transferring the method to a receiving lab, with the key outcome being equivalent analytical performance between them. When we talk about global regulatory expectations, we're looking at a framework that guides how pharmaceutical quality is maintained worldwide. The ICH guidelines set the stage with key documents on validating and developing analytical procedures, managing quality risks, and maintaining robust pharmaceutical quality systems. Alongside these, the USB outlines requirements for transferring analytical methods. The FDA adds its own expectations, emphasizing data integrity, process validation, and current good manufacturing practices. Finally, the WHO aligns method transfer principles with good manufacturing practices, ensuring consistency across the board. When we talk about transferring analytical methods, there are a few key types to keep in mind. Comparative testing means both labs analyze the exact same samples, side by side. Co-validation involves the receiving lab partially or fully revalidating the method. Method verification focuses on confirming critical performance characteristics, and revalidation is necessary whenever there are significant changes to the method. Of all these, regulatory bodies tend to prefer comparative testing the most. It's the most widely accepted approach. When we're transferring an analytical method from one lab to another, there are some key prerequisites on both sides. The transferor lab needs a validated method, an approved procedure, and solid historical data showing consistent performance. On the receiving end, the transferee lab must have qualified analysts and instruments, a controlled environment, and approved standard operating procedures. Plus, we need all the right documentation: the transfer protocol, validation report, method SOP, and a thorough risk assessment. These elements together set the stage for a smooth and successful transfer. Let's talk about the tools we use to assess risk. 3 key methods stand out: FMEA, the fishbone diagram, and the risk ranking matrix. Each helps us identify variables that impact analytical performance. For example, instrument differences and sample handling are high-risk factors, while analyst skill, reagent variability, and environmental conditions fall into medium risk. This kind of risk assessment is crucial during method transfer, guided by quality risk management principles like those in ICH Q9. Understanding these critical risks helps us maintain quality and consistency. This slide outlines the key components of the method transfer protocol. First, it defines the scope, which methods are included in the transfer process. Then, it clarifies responsibilities, splitting them between the transferor and transferee laboratories, with quality assurance providing oversight. Next, it lists the materials involved, like standards, samples, and reagents. The experimental design covers the number of analysts, runs, and replicates needed. Finally, acceptance criteria must be predefined and backed by scientific justification. This framework keeps the transfer clear and reliable. In this study, we're comparing different testing methods across several key sample types: assay samples, impurity samples, and dissolution samples. To make sure our results are solid, we run multiple replicates, usually 6 separate preparations, handled by 2 different analysts, sometimes spread over multiple days. We're focusing on 4 main comparison points: assay, impurities, precision, and accuracy. This design helps us understand not just what the results are, but how reliable and consistent they really are. Here, we're looking at a comparison of assay results between 2 labs, the transferor and the transferee. Each replicate shows how close their measurements are to the label claim, hovering right around 100%. This table highlights the consistency between the labs, which is crucial when transferring methods or validating processes. It's a solid example of how precision and agreement come together to build confidence in the data we rely on. Now, let's talk about precision evaluation. This is where we measure how accurately our system performs. It's not just about getting results, it's about getting the right results. Precision tells us the proportion of correct positive identifications out of all the positive identifications made. In other words, it helps us understand how reliable our system is when it claims something is true. This step is crucial for refining and improving overall performance. Here, we see a clear example of how statistical evaluation supports method transfer between labs. The transferer and transferee means are nearly identical 99.88% and 99.92%, with a tiny difference of just 0.04%. This falls well within the acceptance limit of plus or minus 2%. Using tools like mean difference, percent recovery, t-tests, confidence intervals, and equivalence testing, we confirm the transfer was successful. It's a solid demonstration of how data backs up reliable lab collaboration. Acceptance criteria are the clear, specific conditions that a project or task must meet to be considered complete. They set the bar for quality and functionality, helping everyone know exactly what success looks like. By defining these up front, we reduce misunderstandings and keep the team aligned. It's about making expectations visible and testable, so there's no guesswork when it's time to deliver. When we're transferring methods, several challenges can pop up. Instrument variability is one. Different HPLC systems and detector sensitivities can throw off results. Then there are column differences, like lot-to-lot changes or aging effects that impact performance. Analyst variability matters too. Sample prep can vary from person to person. Environmental factors like temperature and humidity also play a role. And finally, data integrity risks. Uncontrolled spreadsheets and missing audit trails can compromise the whole process. These are the hurdles we need to watch out for. This slide outlines the key components of the method transfer report. It starts with a clear summary, confirming whether the transfer objective was achieved. Then, it presents all the experimental data, including raw data, chromatograms, and calculations. Next, there's a statistical evaluation, comparing results to ensure consistency. Any deviations are investigated, with their impact assessed carefully. The conclusion states if the method was successfully transferred, or if corrective action is needed. Finally, QA approval marks the formal release of the transfer activity under GMP. Let's wrap up with the key takeaways on analytical method transfer. It's not just good practice, it's a regulatory must whenever labs or sites change. The guidelines from USB and ICH give us the scientific and regulatory framework to follow. Risk assessment should shape how we plan and design the transfer. Comparative testing is still the go-to approach, backed by solid statistics and clear acceptance criteria. When done right, method transfer safeguards data integrity, product quality, compliance, and keeps the business running smoothly. That brings us to the end. For any further support or questions, please don't hesitate to reach out. Thank you all for your time and attention. It's been a pleasure sharing this with you.